My name is Alysha Higgs, and over the summer I worked in the HoloLab on the Mitf transcription factor in zebrafish, improving a disease model. So zebrafish are used as a model because they are able to produce a large amount of offspring approximately every 7 days. They are fast developers. They have a similar genetic structure to humans. They share approximately 70% of the same genes that we do. And 84% of genes associated with human disease has a zebrafish counterpart. Their genome is also fully sequenced, and on average, they are cheaper than mouse models. So based upon these things, they are used to model human disease and are used for drug testing and analysis of gene function. So in the HoloLab, we use the zebrafish to model glomerular injury. And the glomerulus is located in the kidney. It has 3 layers: the endothelial layer, the basement membrane layer, and the podocyte. The podocytes contain foot processes that interdigitate with one another and are anchored by actin filaments. And the interdigitation leaves a space known as the filtration slit diaphragm, and molecules are able to filter through the kidney from the endothelial layer through the basement membrane and then through the podocyte filtration slit diaphragm. And if there's an anomaly in any of these 3 layers, it could lead to necessary proteins being excreted through the body through the urine, which is called proteinuria. And in the HoloLab, we have been trying to develop a drug testing model by inducing this glomerular injury upon exposure to puromycin aminonucleoside, also known as PAN. And in order to make this model, it needs to be able to show recovery in order to show the potential that a drug could possibly affect this glomerular injury. So through previous experiments from our 2 transgenic line of fish, we have seen that there is a strain difference. And this strain difference is between our AB fabfish and our NACRA fabfish. And upon exposure to PAN, it is seen that our NACRA fabfish are more susceptible to kidney injury. So in order to see this, our methods were to use our green fluorescent fabfish and spawn for 30 minutes. And at 46 hours post-fertilization, we would transfer them to a 12-well dish and treat with PAN up until 96 hours post-fertilization. And at 96, 120, 144, and 168 hours post-fertilization, we would perform what is called an eye fluorescence assay. This eye fluorescence assay is used because our fabfish produce a green fluorescent plasma protein in the liver, which then transfers into their bloodstream. And if there is no damage, as you can see with our DMSO, they are able to retain this plasma protein, which means that they are able to increase their fluorescence over time. But if we induce this glomerular injury, as you can see with our PAN, then this protein is able to be excreted through the urine, which leads to this decrease in fluorescence. But if it shows signs of recovery, then that means this plasma protein is able to be retained, and the brightness increases over time, as you can see. This recovery is also demonstrated in this graph. So for our PAN 1 milligram treated NACRA fish, we can see that they are able to fully recover at 144 hours post-fertilization. And for our 2 milligrams per ml treated PAN, we can see that they are able to recover at 168 hours post-fertilization, while our 4 milligrams per ml treated PAN are unable to recover, and this could be because the glomerular injury is too severe. So I'd like to take a step back and look at this strain difference. So as you can see, our AB treated PAN fish has no real decrease in fluorescence at 2 milligrams per ml. And at 4 milligrams per ml, there's a slight decrease in fluorescence. But with our NACRA treated fish, we can see at 1, 2, and 4 milligrams per ml, there's a great deal of decrease in fluorescence, which shows an increased sign of proteinuria. And genomic analysis revealed that there is a difference between these 2 strands of fish with the Mitf protein. So our NACRA zebrafish strain has a truncated Mitf protein as compared to our AB zebrafish strain. And this Mitf protein codes for the Mitf transcription factor. And a transcription factor binds to DNA and other proteins, and it is able to control the activity of the gene by interfering with transcription. So the Mitf transcription factor codes for a whole host of genes, but for our lab purposes, we focused on morphology and diaph1, which is also known as dia1. And dia1 helps to regulate the polymerization and depolymerization of actin filaments, which I said earlier is anchored in the foot processes of proteocytes. And without this proper actin depolymerization and polymerization of actin filaments, there can be effacement of the proteocytes, leading to this anomaly within one of the barriers of the glomerulus, which would once again lead to proteinuria. So in the zebrafish, we did not find dia1 in their genome, but they do have diaph2, diaph3, and INF2, which act in a similar fashion to dia1, in that they control the polymerization and depolymerization of actin filaments. And in addition, a mutation in the human INF2 causes FSGS, which is also known as kidney disease. So in order to further explore the pan effect on the Mitf transcription factor, we performed our microinjection on our AB fab fish. So in order to do this, we would still spawn for 30 minutes, but after the spawning, we would microinject the embryos with a Mitf morpholino or a control morpholino. The treatment period would still occur from 46 to 96 hours post-fertilization, and this time we would take pictures only at 96 and 120 hours post-fertilization. So preliminary results from this experiment showed that we are able to downregulate this Mitf gene, and this downregulation causes this AB fish to be more susceptible to the treatment of PAN as compared to our control morpholino injected fish. And with this, we were able to perform qPCR. And if we take a look at the PAN treatment only, all of this is compared to our AB DMSO. So for our AB PAN, our NOCRA DMSO, and our NOCRA PAN, we see a downregulation of diaph2 and diaph3 as compared to our AB DMSO. But for our NOCRA PAN, we see an upregulation for INF2. And if we compare our PAN treatment only to our morpholino injection PAN treatment, we can see that the graphs don't really look the same. But for our control PAN, our Mitf DMSO and our Mitf PAN, we can see the diaph2 and diaph3 genes look about the same as compared to our NOCRA fish. But the results for the INF2 genes are inverted, in that our Mitf PAN is downregulated instead of upregulated, and our control PAN and our Mitf DMSO is upregulated instead of downregulated. And so based upon this, we can see that the Mitf injection doesn't really mimic the NOCRA strand fish. And from this, we could possibly hypothesize that the Mitf transcription factor does not regulate the INF2 gene. So from this, we were able to conclude that we have generated a method to induce kidney injury by PAN, and we were successful in discovering a model for drug testing. And we were also able to characterize the new model and able to see that diaph2 and diaph3 are genes that are possibly regulated by Mitf. So for future experiments in the lab, we would like to further characterize the zebrafish model and possibly see how PAN actually affects those genes, and also to develop a PAN model system in the mouse, because we're trying to collaborate with the Jackson lab that has a Mitf knockout in a mouse model, and currently there is no PAN mouse model system available. And with that, I would like to thank Pat Schroeder for helping me with all of my work, and all the other members of the Howard Lab, and my funding source, the NSF REU. So thank you, and with that, I'd like to take any questions.
Questions for Alysha?
Yeah.
I'm just curious if your analysis of IFN is interferon, is that what that is?
And is that because you're assuming there's an inflammatory response that leads to that degradation of the barrier?
Is your question about INF2?
Yeah.
We explored INF2 only because of its influences with the actin cytoskeleton.
Questions? Alysha, since the actin skeleton is very dynamic, it certainly affects the podocytes, changing the actin dynamics. Do you see other effects, other phenotypes of the treatment that you do that you know is influencing podocytes? Is it influencing other things that involve actin, wound repair, cell division, other things?
So his question is if PAN influences other things within the actin cytoskeleton, and we only really look at its influence on the kidney. So with that, we can see edema at some times, and then we see the loss in fluorescence, but that's really the only thing we look at. So we also make sure that there's flow past the kidney, so we know that it couldn't be something else.
Okay.
But that's really all we look at.
Questions for Alysha? Thank you very much.
Thank you.